Hi, my name is Linnea.
I'm Bezia.
And I'm Linnea, and this is episode number 14 of the Lemma Journal Club.
What is the Lemma Journal Club?
The Lemma Journal Club is the show where we read every paper to tell you if you need to read it. You probably do.
We have a terrific paper for you today. The paper is Convergent Flow-Mediated Mesenchymal Force Drives Embryonic Foregut Constriction and Splitting. The first author is Rui Yan, and the last author is Clifford Tabin, or Cliff Tabin.
So, Bezia, why did you pick this paper?
Because I met Rui back in 2022 at Chicago's Society for Developmental Biology meeting. I've met him a lot since then at different conferences. He's a chicken researcher, and he's a great person, and he does incredibly cool experiments. And he sent me this paper. I contacted him when ChangHee started talking about doing very similar experiments to this paper. I said, someone in my lab is thinking about doing these experiments. I kind of think you've already done them. Is that correct? And he said, actually, we just submitted this paper at the beginning of January, and he sent me a preprint of it. And I skimmed through the preprint, showed it to ChangHee and was like, oh yeah, don't do this project. And I've always wanted to read this paper in depth. It's a monster of a paper. It's 12 pages long. It's 8 figures long. I'm very happy that I finally read Rui's paper in great depth, but I am sorry that I dragged you into this.
I don't think it was a waste of time, but man, was it a long paper. And I didn't even know that papers could be this long. So it makes me wonder about the history of this paper, because it's such a high impact vision written paper that makes me think that it was started out at Nature.
So
but
interesting.
It's so long that maybe they started at...
we have answers to that.
Yeah. Okay.
Because I both know the authors, or, I kind of know them, and the Nature Communications publishes their review process online.
Okay.
So for some reason, Papers, ReadCube, was very nice to me this time and gave me all the supplemental information and all the movies inline as I was reading.
Really?
I don't know what it is about Nature Communications upload to Papers, but it's fantastic. It's exactly what we've been talking about.
Oh my gosh, I have the absolute opposite experience with this paper.
Oh, really?
Because I read it on the web browser.
Oh.
Because I was mostly reading it on my phone while taking care of our infant.
I see.
And the movies are not in line. The supplementary figures are not in line.
Yeah.
And with 21, I got to supplementary movie 21 in the text. I don't know if there were more, but with that many movies, I was not going through and finding them and connecting them back.
Turntables have turned. This conversation has gone exactly the other way. I've been like, oh, there are 35 supplementary movies in this figure, in this paper. Screw that. And you'll be like, oh, actually, I saw them all in line at PNAS. Yeah, it's finally turned around the other way.
Okay, so in the PDF, they somehow did it. Okay, anyways, we're going back to that conversation.
The paper was originally submitted to Nature Communications.
Okay.
I know that because Rui emailed me at the beginning of January saying we're about to submit this paper. And this received date at Nature Communications is 14th January 2025. So this was originally submitted to Nature Communications. It's not bumped down from anywhere. In the reviews, there are 5 reviewers.
Okay.
I would normally insert a curse word there. Your mom doesn't like that I curse so much on this podcast. So there are 5 reviewers for this. One of them is like a sub-reviewer to another of the reviewers. So they've acknowledged that one reviewer is a PI and the other reviewer works for that PI.
Okay.
And it's like an official mentorship thing. All of this, the inline videos where I can just click around the supplements, the being able to see the reviews, having so many reviewers, it only went through one round of review. It was a giant review, but it only went through one. Made me really like Nature Communications. I understand it's paid for, that it's an expensive thing to publish in, but you get to put out a paper like this. The only problem that I have with this is I don't, there's, I think, 3 figures in the whole paper where the caption to the figure is on the same page as the figure. Less than 50% of the figures have the captions on the same page as the figure. That's not the editor. That's not the author's fault. That's the editor's fault. Why am I paying you, Nature Communications, if your graphic design is going to be so deleterious to my reading effort? Okay, that's enough of that. Should we talk about the paper?
Okay, I have some other complaints in this area, which I'm just going to get off now because
get them off.
One is that I read this online on my phone. Great experience. I mean, besides not having everything in line, it was a pretty good experience for getting through the text and like making it pretty digestible. On the computer, they have ads.
They have ads?
They have ads that like blink on the side of the page if you don't have your ad blocker on. So I found that pretty obnoxious from Nature Communications.
Yeah.
Interpreting the figures was also challenging because they're huge figures. And to see them, you have to click on them and then it takes you to another page and you like then have to read the figure totally separately from the text. And it got us back to this back and forth complaint that we've had before. And so for me, this was a very like challenging paper to physically read.
How is that on your phone? This is the first paper you've ever read on your phone, right? I've never read a paper on the, I mean, I like tried to a little bit for this paper because we just started talking about reading papers on our phones.
Well, so I have read papers for this this podcast partially on my phone before. The very first paper that we read, and then actually the last paper that we did for episode 13, I read on my phone as well. The last paper, again, videos were in line even on the phone, so I could play the videos as I was going through, and that was a nice experience, and also just makes it so I can read without Asya interrupting me.
Okay. The paper is beautifully written. I mean, the prose is so good. I think this is the best writing of any paper that we've covered, which is a little bit makes me sad. I don't know how I feel about it. The paper is not head and shoulders above the rest of the papers that we've read. You know, it's, I would say, a good paper, but the prose is so good. It's kind of like a discontinuous thing, right? Like this is a paper that I would have expected to be written in the 1960s. That was like a landmark developmental paper.
I was not struck by the
You didn't love
writing.
the prose? The abstract I thought was one of the best abstracts I've read in years.
Okay. I mean, I don't know. I was not struck. I think there's a lot for me. I'm totally outside of this field, so I felt a lot of jargon. My developmental biology knowledge is negative, despite being married to a developmental biologist. They talk about epithelial splitting in the second, the epithelial folding in the second sentence and epithelial splitting in the third sentence, both of which I don't have an intuition for.
I was not hit by this while reading this, because this really is very solidly in my field of expertise. But developmental biology's penchant for jargon, for words that are specific to developmental biology, and the belief that they cannot get rid of those words because they need that specification, makes the field completely impenetrable.
Yeah, so I found a lot of the writing obtuse because of this. So tetrapod embryogenesis. I'm sure I should know what a tetrapod is. I'm sure I learned it in high school biology, but I don't have that working knowledge.
Yeah, it's a forelimb is a tetrapod. Yeah. So chicken, tetrapod, zebrafish, not a tetrapod.
Okay.
So sometimes when
but even the significance of that. Yeah. So where evolutionarily did tetrapods branch off from non-tetrapods? I don't know. And how, when they say it's evolutionarily conserved, I don't know how far back that goes and what other species are in that.
Yeah, it's a good point. Maybe I have to rescind some of my good writing, because I knew even when they were doing it, they go pretty hard on abbreviations of tracheal esophageal separation. They go very hard on the developmental geometry terms of dorsal, ventral, apical. And I know that kind of stuff was a big stumbling block for me when I started developmental biology. I remember being back at UCSB and the developmental biologists would say these terms to us and we would just throw stuff at them and be like, can you just speak English, please? Stick to one word for one thing. Don't come up with 6 words for one thing.
Yeah. And so this was a problem I had throughout the paper, was just being a little murky on the basics. And so they are talking a lot about this tracheal esophageal separation, TES, and they have a figure. Their figure 1A is, I think, supposed to orient you for what this is. But they don't have a cross-section of the pre-split. So
I think in the dashed box...
Isn't that post-split?
Well, they've kind of done a 2-for-1. It's moving, the split is moving up. So you have your esophagus and your trachea. They were one tube.
Yeah.
Now they're going to become 2 tubes.
Yeah.
And that is going to go from the bottom, where they're already split, and work its way up. right? And so they've given you a cross-section of a place where there's still one tube and they're about to become two tubes. You see that in the figure 1A, the dashed yellow line?
Yeah, I see that. But then I get confused because so sometimes they're showing this double, this two-tube morphology. And my understanding was this was the splitting was happening successfully.
Yeah.
And sometimes they show just an oblong tube where this pinching hasn't happened.
Yeah. So figure 1H, the chick at embryonic day 3.75, starts as a long tube.
Yeah.
And then you move down the sample. So we're moving down.
Yeah.
Towards the posterior direction, if you want to use their words. And you're seeing, as you move down, you're getting to this pinchy shape and then totally pinched off because now it's two tubes.
But then sometimes, where we are in this moving down and moving up, I was never totally clear on. And so I was never super oriented in terms of what the result meant. So if you don't put the constriction on, then you see more of a blobby shape. And this means that this isn't happening correctly, but I wasn't sure about where I was supposed to be thinking about that in this purple and green diagram.
Yeah.
And so this kind of murkiness hindered my understanding throughout the paper.
I see. Yeah. I think that this is also a difficulty in 3D shape science.
Yeah.
That whenever you're doing science in 3 or 4 dimensions, projecting that down onto a 2D piece of paper in a meaningful way becomes challenging to the point of impossible.
Yeah. I hear that. I spent a lot of time on this paper. It's such a massive paper. If it had been a shorter paper, maybe I could have come back to this first figure after reading and been like, okay, so what is this? Let's reorient. But it's such a long paper that I wasn't able to do that. And the text didn't bring me along enough.
I mean, it's such a long paper that I don't think we could possibly discuss every single thing that I've commented on, especially because I'm commenting on, when I'm reading it, a lot of very small things. I was surprised about the hyaluronic acid staining that they did. And I did a little bit of a dive on that and started talking to people about that result. I don't think we can go through all those little details.
Yeah.
So I want to highlight a thing that I think is incredible and a thing that I think is not so incredible.
Okay. Can I guess what you think is the incredible thing?
Sure.
The constriction experiment? Where they rescue? No. Okay.
That is an interesting experiment. I would find it incredible if the imaging of the constriction device was clearer. But because I can't see the PDMS or whatever they use for the scaffold, I'm not super convinced by that experiment.
Oh, interesting.
Which is quite unfortunate because it is a very linchpin experiment.
Yeah.
But I really need to see it.
Yeah.
There's a bunch of experiments that altogether are all consistent with each other. That being said, I can't really see how they did the physical constriction experiment, right? So they say, hey, the esophagus and the trachea separate because the mesenchyme, the surrounding tissue, is pushing in on them. We can get rid of the mesenchyme. It doesn't separate. We can then come back in with a pushing device and it does separate, but I don't see the pushing device that they've built. And I actually made a comment to myself here. I had been saying over and over again while going through this paper, this doesn't convince me, but I'm not sure what would. This doesn't convince me, but I'm not sure what would. And so at figure 2A, I ask myself, what would convince me about the existence of a necessary mesenchymal constrictive force? Because these experiments don't. And even if done very well, I'm not sure that they would. And the problem becomes in my mind that I can't really envision an experiment where I would solidly say to myself that my internal model of this from now on is that the mesenchyme is exerting a force on the epithelium, which is a very weird thing to be skeptical about, right? But I can't pull
So even if you had seen so I found this experiment, this was my favorite part of the paper, was this constriction experiment and the rescue. And so even if you had been able to see the PDMS device and know so one thing I think would have been useful to report is how wide that device was, right? And to give a sense of where it's pushing on the tissue. But even if that had been really well displayed and controlled, you wouldn't believe this external force argument. Just because you have to dissect off the mesenchyme and too much is changing?
You've done a huge perturbation and too much is changing. You're causing the cells to change their behavior by exerting a force on them in a way that, isn't necessarily
similar to what the mesenchyme is doing.
Yeah, this is the best of the experiments that they do. They do some ablation experiments, which they did it with a Leica laser. It's not a fancy pulsed. It's just a normal 2-photon. They're just boiling the mesenchyme away.
Yeah.
And so who knows what's happening at that point. So the ablation, I'm super not convinced by. The movies are not acceptably good. The movies in general, this is not they have a GFP chicken, but they don't have transgenic, so it's difficult for them to get terrific movies. I want to be able to see the forces. Here's the big thing that I don't understand, not just about the paper, but about biophysics. Here's a big thing that I don't understand. I don't understand the force body diagram, the free body diagram on any parcel of tissue. I don't understand this from a fluid mechanics perspective. If you have a cell, a mesenchymal cell pushing on the epithelium with its hands, where are its feet is kind of my question. What is it pushing off of, right?
I kind of think of it as a bag or like a straitjacket, that it's a stiff thing that's holding in a loose thing.
Sure. It's a self-tightening straitjacket.
Yeah.
Where does the force come from how does a self-tightening straitjacket work? Where's the force being generated from? At some point, you've got to push off something else, or you have to make a whole rope all the way around and constrict.
Yeah.
Is that what's happening here? That there's a whole rope all the way around and it's constricting? That could be. But they're talking about flow of the mesenchymal cells to a location.
Yeah. Well, so at that, I got more skeptical and more, I'm not sure what would convince me of this argument later in the paper for the flows and stuff. But I think the thing that I think supports your vision of this as a beautifully written paper is the description of the dissection of the epithelium from the mesenchyme, and that it immediately ploofs out. I mean, they don't use the word ploofs, they describe it beautifully, but that description really convinced me, because I could imagine doing this. I mean, I don't do any dissection, but I could imagine doing the dissection under the microscope and being like, whoa, that's weird. Why did that happen? And then doing this experiment.
Do they say what time scale it happens at?
They said immediately.
Immediately.
And so that makes me think, I guess it was that text that convinced me more than the images that they showed.
This is the problem that I have with the paper, is the text was incredibly convincing. The figures and the movies, particularly the movies, were not incredibly convincing. And so I feel like I've been a little bit fooled. Now, this is all bullshit, because I know that this is all true. ChangHee has worked in a similar system and found very similar things. But I'm trying to put myself a little bit as an outsider here, because I think that this paper is a good check for my own skepticism.
Yeah.
That I'm trying to think about this paper of if I was coming into this skeptical, what would I be thinking about? Because I actually, this is one of a few times where I get to know the answer at roughly the same time as the people who wrote this paper.
So I had a lot of skepticism later in the paper, when they started talking about the flows driving this constriction. I was just when I looked at the data, I was like, well, that's a way to interpret this data. But I could imagine it's a noisy measurement, and they're pulling out a
on the one hand,
very specific signal that fits very well into their story. And
on the one hand, their PIV is very noisy. Their cellular segmentation is actually kind of beautiful based off of what their data looks like. ChangHee just has the same data and has difficulty making the same kind of plots. But ChangHee has much better movies of a very similar mesenchymal migration towards the epithelium. And the movies are clearly the mesenchyme cells are flocking.
I also didn't get to watch the movies of this paper again because of the way that I read it.
These movies are not quite as... I'm just saying that I really do think that this happens, even if it's...
I just was not convinced by the data that was shown.
So I'm just going to talk about what I thought was really cool, or is really cool.
I got us off track.
Figure 3 is where I thought the paper should start. And that's because figure 3 introduces a system in which they slice a bit of esophagus/trachea, which hasn't split already. And they take that bit and they put it in Matrigel and they culture it. And they get to watch this thin slice split. And that is an unbelievable system, because you get to do single cell microscopy You get to do whatever mechanical controls you can dream up. Like, this is a really beautiful system. And it's a really beautiful system that you don't just have to use for what they did. Like, I could use this really beautiful system with my energy, metabolism, mitochondria, glycolysis experiments, and I would get unbelievable results, right? Like, I kind of hope somebody's already doing that. But if nobody's doing that, this is something that I could do and would be really, really beautiful. Like, this is as good as like neural tube culture as far as imaging is concerned. And this was invented by Rui, right? This wasn't something I think I've ever seen anyone do before. And I don't know, I've only seen a couple people pull this kind of stuff off, of just taking like a totally new way to culture a totally new thing in a way that blows everybody out of the water for what's going to be possible for imaging for the next 5 or 6 years. And so I really think that this is the big thing of the paper. In fact, I highlighted in green like 6 times, and I highlight this and I highlight that.
Yeah,
this is what allows them to do the experiment that you are so impressed by, where they do live imaging of this slice with surgical removal of the mesenchyme in the absence or presence of this collagen gel trough.
Okay, so I liked the one in figure 2A.
Figure 2A. Sure, but this is not they're not.
They're not troughing it. Okay, yeah. I was confused about this G, the actual data here.
Yeah, that's what I say. The text is so convincing. Collagen trough, it does this thing. But you look at the actual picture and, it kind of looks like the mesenchyme is still there. I don't see
I just feel
the collagen trough.
I feel that the 0 hour looks so different between the mesenchyme removal and the bilateral pressure that I have trouble understanding what's happening and what's evolving over time.
Yeah.
Again, this is just me, like, my 3D picture, and now we've cut out a little bit.
I think they just didn't get the right, they couldn't get the perfect image. Again, if you look at my paper that I'm trying to put out, getting the perfect image is bizarrely difficult. So you can know something is true, but telling something that's,
I just have questions of like, okay, so do they remove the mesenchyme? I mean, maybe you read this in the methods. They remove the mesenchyme and then they stuff it in the trough, and that's what causes it to elongate in this initial way?
No, I think the mesenchyme is totally removed.
Yeah, I'm saying, do they remove
they stuff the epithelium in the trough.
In the trough. Yes. Or do they do the dissection like in a trough and pull out the mesenchyme?
I don't know the answer to that. I kind of, imagine that they remove the mesenchyme under a dissecting scope, and then they stuff the epithelium in either an imaging chamber with a trough or an imaging chamber without a trough. And then the trough is supposed to, exert pressure. I don't really see how that works. Again, the imaging would be better. The whole thing, again, in the text is incredibly convincing.
Yeah.
In the figures, I don't see what's happening at all.
Yeah, and it's supposed to be like the control would be chick E3.75 in B, and neither of these in G look like they're undergoing anything similar to what's happening in 3.75.
Yeah.
I was just murky on what was going on here.
Yep, I don't disagree with you. This is kind of, I said, my concern about the paper is that the writing is so good that I'm concerned that I'm being fooled.
Yeah.
And I think if I didn't already have some intuition for the system myself, I would probably be deeply skeptical of this. They do have, this is a tremendously long paper. They have 8 figures. They have like 12 supplemental figures, but it has depth in a way that I don't really care about, right? It has a lot of depth in SHH mechanism and in genetics, and not so much depth in the biophysical, where the force is coming from, where the pressure is coming from.
Yeah. How hard are these experiments to do? Like, how hard is it to dissect the mesenchyme off of the epithelium?
So I don't know for sure, because I've never done this stage. And maybe it's easier than I think it is. But I think this is around, from just like a hand-eye coordination, your hands can't shake at all. I think this is about as hard as it can possibly get, where it's still possible to do under a dissecting scope without a micromanipulator.
Yeah.
I don't know if Rui uses a micromanipulator or not for this, but this is the limit.
Yeah.
Like, I could probably train myself to do this over the course of 3 or 4 days, but it would be rough. This is rough. This is the kind of stuff where I often ask Chan if he's done it before and find out from him how hard it is before I try to get my hands. And this is also the kind of stuff where if you don't have good hand stability just as a human being, this is where you can no longer do experiments. That you have to go find someone to collaborate with. Oh, I guess we're going to pause now.
Okay, sounds good.
Pause time. Okay, so we're back. We had to take a little bit of a break because we don't have daycare right now. So, we're on the baby's schedule for recording. We had just finished figure 3.
Out of 8.
I think maybe we'll have like the Who Says Life Has to Be Neat jingle or something. If you're listening to this and you know a way that we can get someone to record us some like intro music for the podcast or intermission or outro music for the podcast.
Maybe we could ask Noah.
We could ask Noah.
Yeah.
Interesting. I wonder that, he might actually,
I think he would like it as a little side project.
Yeah.
I could credit him.
Yeah. I wonder if we could use something that he's already done. I remember going onto his
SoundCloud.
I remember going onto his SoundCloud and being very impressed. And I wonder if we could just use a little clip. Anyways, moving on into the PIV kind of figures. I really want to harp a little bit on this pressure stuff to see if I can get out of you a explanation.
Okay.
They say, for instance, we hypothesized that if mesenchymal cells converge towards the epithelium, they could generate a mediolateral pressure, potentially driving tracheal esophageal separation. What is the pressure?
Well, I think that they have areas of constriction or convergent flow, and they have areas of expansion. Right? There's this blue and this red area.
So there's areas of density becoming lower and areas of density becoming higher.
Yeah.
So like, for instance, let's say that you're in a less dense area, moving towards a more dense area.
Yeah.
How are you moving yourself?
How are you moving yourself?
Yeah.
I mean, isn't this just a question in tissues? Like, how do cells move around tissues?
Yeah. So like on a piece of glass, like it's actin treadmilling.
Yeah.
And I can kind of bring that out to 3D. If you're surrounded by a bunch of cells, you can act in treadmill. But if your density is lower, then there's no cell. There must be some cells that are not surrounded by cells.
Well, there are these cells that are converging.
Sure.
And they're pushing.
And they're pushing other cells that are converging out of the way.
Yeah. Or they're pushing other, they're pushing. Yeah, there are parts of tissues
transduced to. Where's the boundary condition?
Okay, can I switch our frame for a second?
Absolutely, because I think my problem is a framing problem. So yeah.
So when we were doing our PhDs in active matter physics, we would talk about pressure in these active systems, right?
Yeah, and I would usually shout loudly during those conversations, pressure is not a real thing in active matter.
Right, but you also, I think you just put a video up on Blue Sky recently of this experiment that you did where you centrifuged all of the active system onto one side of the chamber, and then you watch it expand into the other side of the chamber.
And so it's pushing against.
It's pushing against the dense phase and expanding.
Which is eventually, if you follow the force all the way, it's pushing against the polymerized sealant on the side of the glass.
Yeah.
It's pushing against the end of the chamber.
Yeah. So I think similarly, that's what's happening here. You're pushing against a lot of fluidy stuff, but eventually this is there's a rigid boundary, right?
I guess the rigid boundary here is the matrigel that it's embedded in.
Sure, and in a developing embryo, it's embedded in a membrane or a shell.
So this is like what I want, is I want some way to like make force glow, right? Just like I want energy to glow, I want force to glow. Totally useless. But like I would like to see the force being exerted on the matrigel if that's what's happening. Or if there's a contractile ring around the tissue and actually there is no force generated on the matrigel, I'd like to see that. I want to see the force change. So like in liquid crystal experiments, you can kind of do this. You can make liquid crystals where the force changes the birefringence of the liquid crystal, and you can see the force change, and it's really informative about like how does the structure work here.
This was part of Alex Tayar's project with the DNA, like that could get sheared apart and fluoresce, and that could give you like local measurements of the force in these active materials. I think you could definitely do this in the hydrogel, right? You could put in some. There, like I think from soft matter physics, there are probes that you could put in that would light up when they're under force or something.
Yeah, I wonder if you can get the resolution. I wonder if you can get like is it like piconewtons or even smaller, right?
Yeah,
like,
yeah. What are the force scales?
But we're going to ask this a little bit later on for what is the experiment you most want, and I think it's something like that. I think the authors of this paper would be like, this is very besides the point of what we're trying to show here, right?
Yeah.
And I would respond, I think that this is the major point of what you're showing here. An interesting thing right there is they, for the mesenchyme, try to use the cell polarity in the mesenchyme. They try to stain for the cell polarity in the mesenchyme by looking at the Golgi, and they say whichever side of the cell the Golgi is on, that's kind of the quote unquote, I mean, they don't say apical, but I'm going to say apical side of the cell.
Yeah, they draw like a vector between the Golgi and the nucleus or something.
Why? I don't know anything about Golgi. Why are the Golgi on the side of the cell which is the way the cell is going?
Yeah, I have. You asked me this in the car, and I should have looked it up, but I did not. And I got to this point, and I was like, "Oh, this is why Bez asked about the Golgi," and I didn't. I still don't know anything about the Golgi.
I wonder if you did the same staining for mitochondria. In fact, I wonder if I looked. There are some places in my samples where I know the mesenchyme is probably migrating to particular places. I wonder if I can use mitochondria as a marker to see which way mesenchyme are migrating.
Okay, so yeah, I guess you're saying like the thing that you I think we skipped from figure 3 to figure 5 about the convergent flows.
Figure 4 is just this. So I do like, a lot of this is, I feel this paper could have been shorter. The reason why I skipped over figure 4 is like I put this kind of data in the supplement all the time. This kind of stuff is known, it's established. It's good to communicate, but it can be communicated in text or in a single panel. It doesn't need to be a whole figure. I feel like there is, figure 4 has a giant chunk of white space after E and F. Literally a quarter of this figure is blank white space.
Yeah, I do think it could. I mean, it could have gone into the supplement.
They've stretched out because they always planned to submit in Nature Communications. So they wrote the paper they want. Yeah. And that's awesome. I would just encourage them, think harder and write a shorter paper because your audience, I'm not saying write a Nature paper where it's 2 pages, that's too short. But middle ground, think about your audience's time that generally, unless they're super dedicated because it's their field like I am, or because they're trapped into it for a journal club like Linnea was, other people are not going to be able to get through a paper of this length. And I feel like this paper particularly could have put a lot of stuff that's in the main figures into the supplement. If you don't like supplement, that's fine. They could have presented a lot of things in a smaller portion.
Yeah. As a counterpoint, I think one of the things that I highlighted in the abstract that I was like, I wonder how they're going to talk about this, was that they say that these forces are evolutionarily conserved. And that's one of the major claims of the paper, I think. Yeah. And I think that this is the major evidence that this is true across systems.
I think that they say the whole story. They always say, hey, the mouse and chick are very similar. And then they immediately go on to say how the mouse and chick are different, right?
Yeah.
And it was unclear to me while going through the story, is it the same thing?
Well, they say the morphology, they claim the morphology is very similar.
Yeah, the morphology is very similar, but the underlying things are very different. And I thought that was all beautifully done and beautifully described.
If we go back to figure 1H, I guess you're saying these are immunostainings. Are really beautiful.
Everything here is an immunostaining. And it's all fixed. So this is why and what this comes down to is when I was preparing my ASCB poster, I had this beautiful poster and I had the first half of the poster and my advisor looked at my poster and basically said, this is a garbage poster. Half of your poster is just immunostaining. And I looked at my poster and I felt, they're right. I could put this all in as 1, maybe 2 pretty immunostain images. I don't need to throw the rest of this at people. Because, again, I said that I really think that this paper starts at figure 3. Figure 3 is where we're doing this beautiful, mind-blowing stuff that I really think will have legs, that will cause this paper to be cited for the next 10, 20 years.
Yeah, I see what you're saying.
So then they go on to talk about, they treat with drugs and show that...
this SHH in a bead, this SHH inhibitor in a bead in figure 8 C and D, they show a picture of the bead. It's huge.
It's so hard to figure out which figure is figure 8 because the figure captions are never on the same page as the figures. So I have to go find a figure, but now I don't know what figure number that is. I have to go to a different page, figure out what figure number that is. This one was figure 7. So this next one must be figure 8. All right, so I'm on figure 8. Oh my God, that bead is enormous compared to this.
It's a huge bead.
Interesting.
And then they're looking at these velocity vectors that are teeny tiny and pointing in every which direction. And I was just like,
yeah, this is again deeply
even when they quantify, like in B and G and H, they try to quantify some things across many samples, and I am still just skeptical because it's A, such a large perturbation, and B, such a noisy measurement that it seems so likely to just be giving you noise as a result.
I can't agree with you more that, again, the text was very strong, and I believed the text, but I don't feel that the figure really defends the text that strongly. At the same time, I will say, in the same breath, ChangHee does the exact same experiments with a soaked SHH bead and sees cellular migration towards that bead. I don't know if his bead, what I suspect is the bead is the same size and the tissue is just about 20 times
way bigger. Yeah.
And so it looks more reasonable. You have a lot more cells to try to track. And it's very clear. If I just saw this paper on its own, I would not necessarily be convinced. But because I've seen ChangHee's data, I do believe that this is true. I think it will be a lot of fun because ChangHee's paper will get published at some point. And if people have skepticism about this paper, they'll see that paper and their skepticism will start to evaporate. I think it's science working the way it's supposed to for once, which is kind of fun. Like, you look at something, you've done thousands of microscopy experiments. You can only show a couple in the paper.
Yeah.
You say, hey, this is my best evidence. This is what I believe. And I've been looking at this. And everyone's like, oh, your evidence is not very convincing. And then you say loudly, well, you do 1,000 experiments and try to boil it down into 10 pages. But if another person who's not you says the same thing and another person who's not you says the same thing, then you get a convincing argument.
Yeah.
So I think that will be fun. That a lot of these figures I don't find super convincing on their own, except for I know that there exists another person who's done similar experiments.
Yeah. And then so the rest of this figure, we get a little lost in the developmental biology. They're doing something with PCAGGS-SH-M cherry.
Yeah, so basically they well, it's not so important, but they're using this electroporation. So I will say the reason why I hold Rui in such high esteem, other than creating this thin culture tracheal esophagus separation system to study how the trachea and esophagus separate during development. Another reason why I hold him in high esteem is me and Megan contacted him about how he was doing the electroporation of genetic material into his developing tissue. And he showed us some of his protocols that described a little bit of what he was doing. And that very much inspired us to not just try what he was doing, but to try many things. And actually, like, the explosion of techniques that me and Megan created to have a genetically tractable system was largely due to Rui's initial messing around in this figure, in which he's trying to upregulate or downregulate SHH and also have a control side, which is the M cherry side, where he's just expressing a fluorophore. And that there's a bravery about the experimental techniques that Rui has adopted for this paper that I have found deeply inspirational.
Cool.
Well, we're on this figure, the last figure, but I want to highlight something here that's super hard to track down until you read the supplement, which is, why is Maha on this paper? Is this the third or fourth paper now that Maha's on? I think it's the fourth. He's on a third of the papers that we cover. But the reason why Maha is on this paper is they have this very brief idea that when you create this concave curvature, where the trachea and esophagus are starting to pinch, you'll get this kind of negative concave curvature. Mm-hmm. And if these epithelial cells are secreting the SHH, then the concentration of SHH is going to be higher inside the cave of the concave area, right? And so those areas that have this negative concave curvature will be more attractive to the mesenchymal cells. And it will be this self-reinforcing instability of the curvature will get bigger and bigger. The SHH concentration will get sharper and sharper. The mesenchyme will be more and more attracted to trying to pinch off that area.
Cool.
That is cool, It is not supported by any experiment in the paper. The supplementary material that shows this is not parsable to anybody who's not us, right? No physicist is going to read this paper. It belongs in a different paper. And it literally just gets one paragraph before the discussion. It doesn't even get a full paragraph before the discussion.
Panel 8L. So I have to be totally honest that I have lost focus at this point in the paper. Like, I think it's fine to present a toy model at the end of your paper and say, this is maybe how it works. Hopefully there'll be follow-up studies.
But the toy model is not presented in graphical format anywhere in the main paper. Like, you could...
Well, isn't that what this L is?
I don't quite know what they really... Working model of tracheoesophageal separation. SHH secreted from the foregut epithelium induces the dorsal subepithelium mesenchymal migration.
They definitely don't talk about curvature in L.
They don't talk about curvature or anything. But in the main text, this is again the before the discussion, they say, it is plausible that the mesenchymal cells are chemotactic to SHH secreted by the epithelium. As the epithelium constricts, a negative curvature then concentrates SHH to generate a positive mechanochemical feedback, which facilitates epithelial splitting. I mean, I know what it means now because I looked into the supplement, but to test this hypothesis, we developed a theoretical model to quantify how tissue curvature affects the concentration of secreted SHH. Analytical predictions and numerical calculations both indicate there's no difference between math works, guys. The analytical predictions and the numerical calculations are the same thing. They don't back each other up. Both indicate that within physiological curvature ranges, negative curvatures from negative 0.01 inverse microns to negative 0.04 inverse microns can effectively increase local SHH concentration by 10% to 40% compared with a flat interface. It's supplementary figure 13. And then they go on to actually describe the whole theory in the caption to supplementary figure 13, which is very confusing. That's it. And then they have another sentence in the discussion that the exact positioning of the centum at the dorsal ventral boundary of the epithelium can further be reinforced by the curvature-sensitive mechanochemical feedback.
Okay. I don't think that can be a paper on its own. And so
I think if you experimentally showed that this was actually happening.
Sure. If there was somebody who was willing to take up this whole thing, like Rui wrote this paper.
Yeah.
This is an 8-figure paper.
Yeah.
He's done with this project.
Yeah.
He is so beyond done with this project. You have to find somebody else who's going to do the follow-up experiments.
Yeah.
Rui had some collaboration. I can totally see how this happened, and I think it's fine.
I see your argument. I just don't know if it's fine. If you don't have any experimental evidence that this is happening, I don't really appreciate just random theorizing. I do some theorizing very similar to this at the end of my paper.
Yeah.
It's very shoehorned in. I do an experiment to suggest that my theory is actually true.
But I also wonder.
I see your argument. I'll rescind some of my stronger statements.
But I wonder if Clifford and Maha are on some joint grant. And they need to be co-last authors for the grant.
Interesting.
And that might be part of the motivation here for cramming putting together this whole theory and not really talking about it in the paper.
They have, I'm just going to say right next to this statement is another statement. The presence of medial constriction rather than dorsal compression can be partially explained by the substantially greater number of cells surrounding the epithelium bilaterally than dorsally. All right, so throwing out all the geometry words, Lydia's rolling her eyes like never before. Developmental biologists, we talk about how developmental biology is going through a rough time right now where there's not as much money in it as there used to be. Developmental biologists, I cannot emphasize how much we are shooting ourselves in the foot by tying ourselves to this boat anchor of nomenclature that developmental biologists will scream bloody murder before they let go of this nomenclature. And it makes it impossible for anyone else to care. They just close your documents as soon as they see this nomenclature. Okay, ignoring the nomenclature for a second, they say, look, we say the mesenchyme is swooping in to follow this SHH gradient to make this pinch point. The reason why the pinch point occurs in the direction that it occurs is because there's more mesenchymal cells in that direction than in another direction. I think that's really cool. I mean, we were talking about, this theory of Maha's can't be a paper on its own. Fine. I think that could be a paper on its own. That could have been a little sister micro paper, because I think, I said that I think that this paper will be cited for a very long time because of the system that it develops. I think that statement on its own which is kind of just figure 7, could also live a very long time. And yeah, I was just like, there was a couple times, but this was, I think, one of the biggest ones where I was like, I think that is a whole finding that is citable of itself. And I wish that this was chopped up a little bit. But, this is always what we're not reviewers asking them to re-chop their paper. Consider making your paper more digestible when you write these mega papers. You don't have to. Just consider it. Think about the digestibility of the paper while writing it.
Yeah. And I don't believe I was the target audience for this paper. And so.
But I was.
Yeah, you were.
I think I was like the direct
But you did also
audience.
say that this was the best paper, most beautifully written paper that you had, that you've ever read. So
the more we talk about it, the more I'm thinking about that statement of I'm very immune now to the nomenclature of developmental biology. But just reading any sentence here, just trying to read out any sentence out loud to you, I'm like, come on, what the heck does that mean for this person, right? So maybe I'll backpedal a little bit. That maybe I'm just very happy once to not be reading about algae. And I'm like, I understand some of the words today.
Yeah, science is a lot of words.
Science is a lot of words.
So, okay, should we go through our favorite of the questions?
Sure.
Yeah, what do you think is intellectually beautiful about this paper?
So I touched on it briefly before, but I think what is intellectually beautiful about this paper is the experimental bravery of Rui here. And sometimes it's a flop. Sometimes it's meh. Sometimes it's so big that I think will create a field of itself, such as creating this slab system that's being cultured in Matrigel. Or it's so big that it has changed the trajectory of my scientific career, which is his experimenting with how to get genetic information into the chicken at this super early age made me rethink how I wanted to get genetic information into the lung at a later stage. So what I think is intellectually beautiful about this paper is Rui's bravery, and then I really think Cliff Tabin's capability to tell a story. That I've seen this from him before in other papers. I really think that he is a beautiful scientific storyteller for me personally.
Yeah. I think the moment I found the most beautiful was the description of the cutting away the mesenchyme and seeing the epithelial sploosh out. And to me, that's beautiful in the curiosity-driven science. That's weird. Why is that happening? And that motivated a lot of the paper for me.
Yeah, I guess I wonder what the writing process was here of how much it was written by Rui and how much it was written by Cliff. I'm giving Cliff a lot of the credit for the writing because it reads like it was written by an older person. But it could be it's Rui's voice, actually. I'm not sure which one it is. It really reads like it was written by one person, and it wasn't. That's a very interesting sleight of hand. I feel like in my papers that I publish with people, you can really see that it's written by multiple people. That there'll be 2 sentences in one prose and then 2 sentences in another prose. And
you're talking about your own papers?
My own papers.
Well, that's because you can see it, right?
Yeah, but maybe that's because I can see it. That's true. If you could have one thing added to this paper, what would it be?
I think I would have things taken away.
Yep.
I think this is the first paper for which that's really true for me, that it was so large that it became undigestible. And so to take away some data or streamline in some way, I think was my biggest thing that I wanted from this paper.
Yeah, I've been saying that, but again, I will reiterate this idea of using a matrigel in which you can somehow do force microscopy with some sort of embedded liquid crystal. Yeah. I don't think that's a real technique. I bet you it doesn't work because people do forced traction microscopy. And if they could do liquid crystal glowy microscopy, they would do that instead, right?
But you could, well, I don't know, but what Alex did, right, is she had these double-stranded DNA things that when how strong DNA binding is. And so you know how much force it takes to shear that apart.
Did she publish this? Because my question essentially is, did it work?
Yeah, I
remember the strategy. I want to know, did it work?
I think we're going to have to go back through this whole episode and check about what we should be talking about for other people's work.
Because you think it might not be published.
Well, if that's not published, if ChangHee stuff isn't published.
ChangHee stuff isn't published.
I would ask him, I think that it's just touchy, right?
Okay.
For anything unpublished. Martin says this in his standard operating procedures. One of the things is not only do you not know what the motivations of each individual researcher are, but there could be, if this is ever going to be patented, it has to be like the first public. So there's a whole thing that we're just not even.
Patents. Okay. So maybe there'll be a lot of heavy editing to this. So if we end up doing a lot of cuts because of that, I think I'll leave this conversation in so that people can kind of understand why is this so heavily cut this episode.
Yeah.
And if we've done a lot of cuts and this has been left in, this was heavily cut because we had a lot of discussions about work that has not been published yet. That Linnea brings up a very good point. Then maybe that would be problematic for patents. We've created a hell of our own making in which it's impossible to make any progress.
But we are publicly releasing this. And so it's not our work. If it's our work, I think we can talk about it.
Yeah. So then, Linnea, what was this paper about?
So this paper was about the process of creating 2 tubes from 1 tube.
Oh, that's good. Yeah.
And how creatures, tetrapods in particular, which are 4-legged creatures. Four-limbed creatures.
Because a chicken ain't got 4 legs.
Yeah, but we're tetrapods too. So this is how the paper is about how tetrapods during development go from 1 tube to 2 tubes in creating the trachea and the esophagus. And the paper has identified a conserved mechanism of force from the outer layer of tissue that's influencing the branching of the inner layer of tissue. And this is due to SHH signaling. Cell migration by SHH
then we get into
signaling.
The dogma of biology of like saying this is entirely due to one thing or 3 things. Or, they have a tool, they play with it. And then for me, I will say for Asya, why did me, your father, care? Yeah, again, this was a person that I met. I met him over and over again, and I will meet him over and over again at conferences. I thought he was a very good scientist in person. He was very sharing with the techniques that he was using, which really elevates someone scientifically in my mind to like the highest level. Like the thing that can make me think that you are truly an amazing scientist is to be open with how you did something, which almost nobody is these days. And talking to him gave me a tremendous bank of ideas for ways that I could be brave in my own experiments, and really created this big push to master chicken genetics, which I eventually started doing with Megan Rothstein, and has been the start of a major friendship in my life. And I guess a lot of things for me in my life started because of conversations with Rui just at his poster presenting this work. And so I thought it would be good to go through the paper that was the finishing to that work, and we'll see where his career goes from here. But I think regardless of where his career goes from here, the experimental platform that he has developed will live on for a long time now. Yeah.
Okay. Anything else that we need to talk about with our listeners?
We'll do episode 15 next week,
hopefully,
which they've voted on, or maybe they haven't voted on. If like less than 3 people have voted,
then we'll just pick our own paper. I'm just going to
pick whatever.
I think a short paper might be nice.
I have just like such a long list of papers that I really deeply want to go through now.
Okay.
I'm not going to ban 12-page papers, but I complained a lot about your cell papers being too long, and I apologize for throwing this at you.
No, I think it made me appreciate your complaints about the last papers more, because I think those papers were as far afield for you as this one was for me. And it was painful to get through.
Medium-sized papers, people. Publish medium-sized papers.
Or short papers.
Yeah, my problem with the short papers is just we've tried to write a couple short papers now.
And there's still a lot of work.
It's still a lot of work, but also like you get into this thing where you start cutting genuinely critical information because of this arbitrary word limit. That like there can become a point where you are damaging the science by trying to make it too short. So you want to think really hard about what is the shortest paper that I can make, and then not make it shorter than that for no reason.
Yeah. Okay, great.
Yeah.
Thanks for a good paper and for sharing the inspirational notes about experimental bravery. I think that should be the title of the podcast.
Experimental bravery. Okay.
Yeah.
Okay.
Bye.